I don't want to be too old. Okay. So we are starting a potential HCA flagship, HCA Asia flagship, and it's about the skin. And maybe I would like to give some background as to the skin research and why it's important, and what are some of the current status, and it'll be presented by students of Professor Hyunje Kim. And then I'll also share a bit about some of the skin activities that we're doing in Singapore. And maybe we'll open it up to everyone. Love to hear from you what kind of skin activities that you guys are doing or have access to, and how we can start to build the atlas. So Hyunje, do you want to say something? Or
Thanks for inviting me. My name is Hyunje Kim, the assistant professor in Seoul National University, and I also work at the University Hospital. And we actually in our lab, in our institute, we have built up some skin disease atlas. So we wanted to share how we constructed the atlas and how we can make the group work in here. So thank you.
Okay, then maybe we'll invite the students, Soyeon and Yuna, to come and share some of what's happening in Korea. So please.
Thank you for the introduction, and I am Una Choi, and today I would like to share our lab's experience and thoughts on the skin research. So these are the table on contents. I'm going to introduce the first half mainly about the samples and metadata, and Soyeon will elaborate from tissue processing and introduce her work on skin atlas and spatial studies. For the first, I would like to start with the public data sets used for the annotations and validations for our findings. Based on the insights from Professor Jong-eun Park from KAIST, we are currently gathering skin public data sets. For the inclusion criteria, we are considering skin biopsies, full thickness skin surgical specimens, and PBMC data. And those are the exclusion criteria because they are known to have different proportions compared to conventional tissue biopsy samples. Now I want to discuss important considerations when building an atlas based on our lab's experience. At the clinical level, I would like to emphasize three factors. First, patient's confirmed consent. Second, sample selection. And third, the metadata. To start off, regarding the patient confirmed consent, we highly recommend obtaining the IRB documents that state consent for the third parties to have access to patient clinical and sequencing data. This will enable you to share data to global initiatives such as HCA. Second, there should be a wise decision for the sample selections. It is important to consider various demographic factors for diversity, and including the self-reported ethnicities and age groups. As an example, I would like to share the diverse age distributions in our lab's atopic dermatitis studies. Our lab had established comprehensive data sets from 5 different projects. We aim to include a broad range of ages by collaborating with the pediatric department to enroll younger patient groups, as well as collaborating with veterans hospitals to recruit the older age patient groups. The third aspect that I would like to emphasize is the patient metadata. I know that the more the better, but excessive data can be counterproductive, so I want to emphasize a few. First will be the location of the biopsy, because anatomical differences in skin affects the cellular compositions. Like, for example, as you guys all know, the skin on your palms is much softer than the back of your hands. Moreover, disease severity scores conventionally used by clinicians are essential. This will help us understand the disease biology itself and also the progressions. Lastly, I want to emphasize the information about the ethnicities and residence. as the skin is influenced by various social environmental factors, including lifestyle, diet, and microbiome.
Yeah, so I'll move on to the sample processing and data generation parts. One thing that we want to stress is the tissue sample processing steps. There may be variation depending on the tissue type, which may be full thickness punch biopsy, excisional biopsy, or suction blister, as Yuna mentioned earlier. And also the data composition and quality can vary greatly depending on the dissociation method. The different methods would be enzymatic dissociation, culturing the tissue for a few hours for emigrating cells, and sorting or enrichment of specific cell populations of interest. And our group usually uses the enzymatic dissociation method of punch biopsies using LiverSTL for a short incubation time of 1 hour. And we also do not use the samples freshly because over 90% of the cells would be keratinocytes then. So we freeze the cells and then thaw them before generating data to increase the cell diversity. And to profile the cells, as mentioned, cell storage and thawing steps are also critical. I wanted to share the results of the PBMC storage tests that our lab conducted, where PBMCs were isolated and divided into different vials and stored in liquid nitrogen or minus 80 degrees. And then the viability and cell number were checked every year. And as you can see, the viability dropped to less than 10% when the cells were stored at minus 80 degrees. And I would expect tissue-dissociated cells to be even more sensitive. So it's important to store cell stocks in liquid nitrogen and take caution when handling them so that the sample temperature is maintained. And as everyone probably knows, when thawing the cells using pre-warmed media and thawing rapidly but gently are critical. And strain the cells if necessary, especially for tissue samples. We recommend to not refreeze the cells more than twice, and ideally to profile the samples within 2 years. Many commercialized data generation platforms are available, and some non-commercialized platforms could be used as well. The different platforms have their pros and cons, and selecting certain platform could depend on the specific research objectives, budget, and data compatibility. But regardless of the platform chosen, ensuring data quality and consistency across different sources and integration methods will be needed to combine the data for downstream analysis. And our group is currently building an inflammatory skin disease atlas of pediatric and adult atopic dermatitis, plaque and pustular psoriasis, hidradenitis suppurativa, and vitiligo. By building atlases, we can unravel the pathomechanisms of skin diseases with high clinical unmet need, define disease endotypes within and across the skin diseases, characterize conditions with overlapping phenotypes, improve treatment selection, and expand therapeutic applications. So a brief overview of each disease and the number of samples. AD is a Th2, Th22 type disease, and pediatric and adult samples were included. And Asian AD has been reported to differ in its phenotype from European American AD. And for psoriasis, both the plaque and pustular types were included, where Th17, IL-23 axis, and IL-36 axis are known to be key. And HS is a rare disease having complex immunopathogenesis mechanisms with high clinical unmet need. And vitiligo is a deep pigmentation disease where CD8 T cells, interferon gamma, and regulatory T cells are thought to play some roles. So we have generated in-house data sets across these diseases for a total of approximately 300,000 cells for tissue and 600,000 cells for PBMCs after QC. And I'll be sharing just a little bit of the results for keratinocytes, which were annotated into 7 different subsets. Some changes in granular spinous and basal keratinocytes were observed for frequency. And as expected, the proliferating keratinocytes were increased in psoriasis, as it is a hyperproliferative keratinocyte disease. And on the right, by using gene set scoring, using genes induced in cultured keratinocytes stimulated by individual pro-inflammatory cytokines, I inferred that in addition to the IL-17A and 36G scores, IL-1 beta and TNF scores are also elevated in GPP, suggesting its highly auto-inflammatory signatures. And I'm doing further downstream analysis of the data and looking into other immune cells, including T cells as well. And we aim to construct an inflammatory skin disease atlas at a single cell resolution and identify both distinct and common pathomechanisms and pathogenic cell types. In addition, we hope to suggest possible therapeutic targets and predict clinical responses to treatments. And we also have conducted a pilot study to generate some spatial data for these diseases using the 10x Xenium. Our plan is to build a spatial atlas to integrate with our single-cell data. Our strategy is to use 3 different platforms, the GeoMx, Xenium, and Maxima. The GeoMx is not single-cell resolution, but covers the full transcriptome, so can be used for discovery. And for Xenium, we have used the pre-designed skin panel, and we're in the process of designing a custom add-on panel of markers that we're particularly interested in. And finally, we aim to validate our transcriptomic findings at the protein level using Maxima for cross-validation across these different platforms. Yeah, so this is our group. And if anyone has any questions, you can always email us. And yeah, Jay.
Well, maybe I'll finish my part and then we can open it up for questions. So I'm speaking from more Singapore efforts. In Singapore, we recently acquired a funding to study skin aging of Asian cohorts. And the idea is to do large number of photography and skin color assessments, skin elasticity as a general easy to acquire phenotypes and measurements, but only the subset of that we will take punch biopsies to do molecular characterization, and that could try to do multi-omics. So not only transcriptomics, but metabolome and lipidome as well. And for subset of that, we'll then do some skin modeling in vitro. So it's a huge project and involves multiple groups. I'm just representing the omics aspect. And so I can't speak for everyone, but this is just for us to get things rolling. And the key idea is Singapore has this funding. I think Korea has really great programs and access to this tissue. I have friends in Japan who also have very large collection of skin. I just met someone from China who have also access to a large number of skin. And this really sounds like a perfect opportunity to start standardize some of the methods and to build the atlas together. So I'm going to talk a bit more about spatial. So resolution does matter. So what you see on the left, to your left, is the Visium-Xenium comparison. And clearly there are key differences in the granularity. And on the right-hand side, you have the Visium HD. So having higher resolution is going to be extremely helpful. So I just made this table. It's very rough, could be a bit outdated, but there are generally 2 spatial platforms, either NGS-based or image-based. So if you go to the NGS-based, there's 10x Genomics, Visium HD, or NGS StereoSeq. StereoSeq has recently released version 2, which will allow FFPE sections, but it's using random priming. So you're going to capture more total RNA, but you may also sequence ribosome RNA. So there's a give and take in terms of the sequencing throughput. Visium HD right now is compatible with FFPE, but then it's probe-based, so you can only target the 18,000 protein coding genes. If you're interested in non-coding RNAs, microRNAs, you won't be able to do that. And also because it's probe-based, you're not able to understand the genetic profiles because you're sequencing the probes. So 10x has recently announced that maybe early next year they will release the cDNA, so RT, reverse transcription-based Visium HD, which will allow us to also capture the genetic variants through direct sequencing. So yeah, these are kind of sequencing-based. Of course, cost is another issue. There's an image-based, there's a Xenium, Cosmex, or GeoMx. And again, these are just for RNAs. We can talk about the protein-based in other time, but basically Xenium and Cosmex now can reach up to 5,000 gene panels. But of course, more genes you probe, less sensitive your data will be. So sometimes you need higher sensitivity to make the UMAPs very clear and distinct. And I'll talk about that a little bit later. So more may not necessarily mean better. Okay, so this is something for us to think about. And if you're going to use a skin panel, how are we going to standardize this across the HCA Asia community? And I think Hyunje's group have already started building some of these custom panels, and perhaps this is something that we can consider for the broader community. So some of the work is a bit old, but still preparing the manuscript for this. So on the left, again, is a Visium SD, so standard definition, just looking at different disease states. And you do see a very distinct signatures of this S100 genes as they're being in the more severe condition of atopic dermatitis. On the right, it's actually aged skin versus the young skin. And when you look at single-cell RNA sequencing, one thing that really pops out are how heterogeneous your fibroblast is. And I think this is one of the key aspect of the skin is the abundance and the heterogeneity of the fibroblast. And in fact, this fibroblast 2 at the bottom right is actually age-associated. So you only see that cluster enriched in aged skin, and they're relatively absent in the young skin. And of course, there are efforts to build fibroblast atlas, because fibroblast is everywhere and every organ has one. So what's so unique about fibroblast? And today we heard in the brain, astrocytes, which are like the fibroblasts of the brain, are heavily involved in Parkinson's and of course Alzheimer's as well. And we all know stromal cells in tumor also play a key role in drug resistance and tumor progression. So we could even consider more cell type specific aspect of the skin, like fibroblast, and how they are distinct across different disease and different age and different ethnicity, for example. This is also this is a very fresh data. We are trying Visium HD in the lab. The first experiment was not great, but we are seeing some interesting results. So this is in collaboration with one of my colleagues in A-STAR, and you have alopecia areata, so it's baldness, and they are treated with JAK inhibitor, and you have responder and non-responder, and we have responder, you have higher accumulation of immune-associated genes are highlighted, so there are a lot more immune infiltration in responder patients. And if you look at broader gene signatures, indeed there are more immune activation in responders, but you have more collagen maintenance here in non-responders. So there is a close relationship between immune cells and stromal cells, and that cross interaction in the skin is quite critical. I didn't mention it, but even in atopic dermatitis, we do see a decrease in fibroblast function as the disease progresses to a severe condition. And if the patients are undergoing a treatment and they have restoration of healthy skin, they have increase in fibroblast function, right? So it makes sense. And so there's interplay between the keratinocytes, the epidermis, and the epithelials, the interactions with the immune cells, interactions with the stromal cells are critical. And I think skin is really the perfect organ to study this kind of interaction, especially in a spatial context. So my last slide is not my data. This is actually from 10x Genomics. They actually provide this type of data for us to play around with. This is human skin melanoma, FFPE, xenium using 5K PEM tissue panel. And on the right-hand side is tumor, on the left is kind of tumor adjacent. What you see here in the UMAP is, I would say, quite distinct clusters of these immune cells and cancer cells and epithelial cells. And you don't get this kind of UMAP when you use Visium HD. So the Visium HD is extremely noisy, and it's very hard to distinct clusters because you have too much data and not so much sensitivity. Here you have 5K panel, and we can appreciate very distinct cell types that are represented in even in the epidermis layers of epidermis. And on the right, you get nice clusters. So I think it's just something for us to think about when we decide which platforms to go for, is do we want to do everything and try to profile as many genes as possible for discovery sake, which is important. And sometimes we need those gene sets to do gene ontology analysis and gene set enrichment analysis. Or do we want to focus on gene panels where we have 200-300 really well-defined skin panel? Or do we want to go 5,000 pan tissue? We sacrifice a little bit on the sensitivity, but we can get broader coverage of the gene profile. So we don't have the answers now, but just something for us to think about. So yeah, this then comes to the discussion points, and this is generously provided by Hyunje's group, and we can kind of go through this one by one and get your opinion on this. So before we start, any questions about what So-young and Una presented or what I presented? Okay. So I know we said skin and microbiome, and I know there's a microbiome expert here in the room. Of course, we're open to study microbiome. Of course, there are microbiomes on the skin as well as microbiome in the gut. There are groups in Singapore that are looking at microbiome of the skin by just doing the tape analysis and doing mass spec and things like that. So there are ways to do microbiome in conjunction with the molecular profiling of the skin. But another key aspect that we should really emphasize is, we're talking about Asian skin. What is it that is quite unique with Asian skin than other ethnic groups? And I think there are a few physiological phenotypes that are clearly different, but then why is that different? Is it lifestyle, or is it genetics, or is it the diet? So we actually don't have the answer. And ultimately, we want to kind of build the initial atlas to garner interest from various funders. And one of the funders could be industry partners. And I can assure you there are many companies like Procter & Gamble, Shiseido, and many other skincare companies are very, very interested in, especially the skin atlas, both for health reasons, but also for potential therapeutic or prevention reasons. So I think we do have a really good opportunity to scale and diversify, and hopefully you'll get your support in this. Hyunje, anything you want to say? Good? Yeah? Good. Okay, so I kind of mentioned like what is the final goal of this, short-term, mid-term goals. So let's go with the why. Like, why are we doing this in the first place, right? Can someone help me? So I'm not a dermatologist, but Hyunje is, right? Formally trained, even not. Yes. And of course, in line with what we were talking about yesterday in this room, we talked about disease. And disease is also a huge spectrum, and it's not categorical. And you also heard psoriasis, dermatitis, many other conditions that can be quite severe, which largely affect the lifestyle and quality of life of these individuals. And to a certain point, if going to extreme cases, it could be very severe. And there are skin cancer as well in that aspect. Traditionally, in Human Cell Atlas, we try to build reference map, like healthy reference. But again, with the introduction of spatial technology, we have the opportunity to profile disease samples. So maybe I'll pose the question to the audience: should we do all this disease for HCA Asia and also build a healthy reference, or should we focus on one disease but try to profile as much samples as possible? Any thoughts? Yes.
So what's the comparison between if you just do a healthy one, so it's like a developmental questions, right? How this developments, or if you compare something such as UV damage or something, is it more relevant to founders or to a broad society? Because UV, well, any damage for repairing process, can be interesting. And what kind of campaigns or treatment to help with the repairing process. Yeah, can be ultimate goal or industry interest with that. I think UV damage in Australia, in other countries, especially Europe, and it's a huge a cause for cancer, skin cancer. If you can prevent that, it could be an interesting topic, and it's just like a data collection. Use that to to plot into your healthy ones to see what kind of population, what the cell population changes. What's the repairing mechanisms? Is there potential target for that?
Yeah.
So just throw out some really random.
Yeah, absolutely. And I think there's a notion of, depending on the region of your skin, because skin is the largest organ of the human body, but you're exposed to UV in different ways. So in Singapore, there are efforts to collect skin samples from 3 different locations, from the UV exposed forearms, underarm, and buttocks. And buttocks being supposed to be UV unexposed control. And the question is whether UV can play a role. And this is Singapore, so there's a lot of sunlight every day of the year. And they do have generally a lot more UV exposure than any other parts of the Asian countries. But of course, when we go to Australia and Europe, you have much more fair skin. And so question of pigmentation, the level of pigmentation was raised. And in Asia, there's also a gradient of pigmentation from South Asia, like in India, to Southeast Asia, Philippines, to East Asia, where Korea, Japan, China. And so how does that play a role in response to UV damage, and how that alters the process of aging? I mean, these are questions that have been around for many years in the skin field, but I think if you can understand this better, again, it could be very attractive to some of the industry partners as well.
The UV exposed, UV non-exposed is very, very important. We have to annotate those informations, and then we cannot do everything because we have limited resources and a limited manpower and limited fund. So I think that the most important discussion point today is where do we have to be focused on. So as a clinicians, I'm keep persuading the clinicians to send us a lot of kind of samples, but always that they are ask me back that, so where, how many, so why? So the mission in this field for me is to collect as many samples from the clinical part, especially in South Korea, strategically. So I just wanted to ask you that in what point we have to kind of the focus on in a clinical field, and then the profiling field and the data integration field like that.
Yeah.
Hi, I'm Bimbi Lai from Peking University, China. I collaborate with the colleagues. They are at the Department of Dermatology, and they focused, or they did the surgery on the EMPD and the AM, acromelanoma. So actually, we have quite a lot of that samples.
Melanoma.
Acromelanoma.
Acromelanoma.
Yeah.
Okay. Do you have, how about other inflammatory samples, like dermatitis or?
Other diseases?
Yeah.
Yeah, I think including your.
You have access to those samples? Or?
I mean, it's depends.
Yeah.
Yeah. Because I'm not the clinician.
Yeah. Okay. Yeah. So again, I agree with Hyunje. Is where should we focus? Because we can't do everything, but at the same time, what kind of most impact that we can create and value we can bring, not only to our research but to the broader community.
Yeah,
And yeah, there's a hands back up.
I'm a rheumatologist doctor in Taylor, south of Taylor. As for our rheumatology disease, many patients have skin rash, such as the lupus has the specific facial skin rash, and the myositis still have specific rash, and the scleroderma has skin fibrosis. So we take some of the skin biopsies and do the single cell data analysis. For our doctors, we want to use the skin biopsy to predict the disease outcomes and the drug response, such as scleroderma is a disease characterized by multiple organ fibrosis. If we can use the skin fibrosis to predict if the patient can develop lung fibrosis or renal fibrosis, it will benefit the patient. So we want to use some of the fibroblast subtype to predict. And for other diseases such as myositis skin, some myositis can develop to tumor, and some myositis patients will not. We want to use the skin cells to predict if some patients can develop or to tumor or not. We can take a lot of the rheumatology skin sample.
Great, that's excellent.
That is very, very important questions that every clinicians in this field wanted to know. So my question is always that to solve that questions, we have to have labeled, we have to have the answer first, like the longitudinal lab samples, and then we know the patients.
Can you use your microphone, just in case there are people on the Zoom?
So longitudinal biopsies, and then some patient kind of some patient progressed, some patient didn't progress, and then it's a simple way to kind of find out some subsets or the gene modules that is associated with the data. But the current status is that we are not doing the kind of the scheduled longitudinal biopsies for to the patient. So always that is the point I think that we have limitations on. So do you think we can predict the patient trajectory based on the early time point single cell spatial data?
I would love to.
I think it's possible. It's possible, but definitely somebody. I think that the pharmaceutical companies already know that. So, but academically, I really wanted to kind of know that point. Yeah.
Actually, we use the back sequence data from the skin fibrosis. We can predict the lung fibrosis and the disease, the patient's outcome.
So you can use bulk RNA sequencing to predict what the outcome may be.
Yes.
And how is it just based on biomarkers?
Gene signature.
Gene signatures?
Yes.
Yeah.
But I think single cell may be better.
So you have some use case or examples that you can showcase that molecular profiling can help to predict the outcome of a patient. Just an example is available, right? Yeah.
We use a small cohort to have some predict significance,
Great. Yeah.
but not in the large cohort.
Yeah.
Yeah.
Great. Yeah. And these are, again, lupus patients?
In scleroderma.
Scleroderma?
Yes.
Derma? Yeah.
Yes, for scleroderma, the lung fibrosis is affect the patient outcome more than the skin fibrosis. For doctors, we focus more on the lung fibrosis.
Yeah.
Yeah. but it's hard for us to take the lung skin or lung biopsy.
Yes. Yes.
So I have one question about the healthy samples. So if we are going to collect the healthy samples from diversity of patients, how healthy does that patient need to be? For example, is it 100% healthy, or just other patients in dermatology, or just other patients in plastic surgery, but we could collect the tissue samples from them? So I think that could be one point of discussion. We are going to, if we are going to collect the atlas and draw the picture of the healthy sample, healthy people. So do you have any ideas or opinion?
Anyone?
How we can define healthy?
The UK Biobank, they use different age groups. So yeah, in China, we're going to do the same. So we just define the people's like age. So just just do a blanket like from after you're born and just go upwards until like 18. Like you have different time gaps for each. So we don't define healthy. We just define the age groups and the comparison, the differences. Because you can't define healthy, you can't define aging. It's just cell changes, right? Well, that's the kind of strategy you're going to use in China for this project. Yeah.
If I can comment on that, I think, we need more data to understand the mean and the deviation of what we think is healthy. Of course, in IDA, we had exclusion criteria where if you are under medication, or if you have been vaccinated, or if you had infection in the last 2 weeks, things like that, we would exclude these samples into our atlas. So we can define such exclusion criteria, which means for the skin, we may need additional metadata, like photography or elasticity measurements. So these could help to then come up with some standards to say this we think is within the range of healthy and not healthy. And of course, all the metadata regarding male, female will have differences on those metrics, but also age as well. So what more and more we break down into these groups, statistics go down, the power of statistics go down. So therefore, we need more samples. That's the point. Go ahead.
Hello, I'm going to talk from South Korea. So I think it would be very important to build a uniform format of metadata to integrate some data, because there are many various data from different institutes. So they use the specific their own metadata. So I think it would be very important to integrate them. So I wonder if there is a specific uniform format of metadata, and how do you think about that? And so how can we build a uniform format of metadata?
Excellent point. And every HCA meeting we go to, metadata, and metadata. Yes. So you're absolutely right.
As a clinician point of view, personally, I think that the current disease annotating system is wrong. Think about it. Atopic dermatitis, what does that mean? The atopic means that aberrant immune response to nominal antigen. So immune response is happening, but dermatitis is just skin inflammations. And then the clinicians, historically, clinicians were defining the disease following their phenotype. Very low resolution data that because of, so it's time to change the nomenclature, I think, that because we human beings have good tools. So I personally, I cannot agree that on the clinicians that this is this disease, this is this disease. So I think that that we have to have a same vision to kind of reclassify the disease annotating system. But the hospital guys don't like it. But
They never have to change that's their job.
because as an immunologist, I think that after development of dupilumab, they strategically got approved from the AD field and expanding the drug, the applications to the allergic rhinitis and the asthma. In terms of the immunologist point of view, those three kind of diseases are same. Just the target tissue, just the same phenomenon, but we are kind of stratifying atopic dermatitis, asthma, and the allergic rhinitis. I think that is the IL-4 receptor mediated disease. Okay, so
Well, excellent. I guess I understand the challenge of changing the whole clinical nomenclature, but what we can do is to raise concerns through biomarkers, saying, hey, these are new biomarkers or signatures of genes that we can help the clinicians to make better choices, for example.
Another the example is a melanoma. So when I was a resident 10, 15 years ago, melanoma can be classified in 4 categories: lentigo maligna and nodular acral-lentiginous melanoma. But those classifications cannot help the patient at all because they do not give any information to the just for the happiness for the clinicians. The different phenotype. So in the cancer field, the cancer annotation is being changed from the mutation based, like the BRAF mutation having melanoma or not. So I think that we have to do everything from the beginning together.
Yeah. More omics and more molecular characterization. Yes.
So I think each organ has its own important clinical factors. So maybe the skin UV is very important for clinical factors. That I think so. So I wonder for skin data atlas, what is the very essential point for interpretation of data? So it may be UV and the other things. So I wonder. Yeah. So, what is the important clinical factor for skin atlas disease?
So, what is the most important clinical clinical benefit?
Yeah. So if we have to build a uniform format of metadata, then which should be included for the metadata?
I see. Yeah. What do you think?
Yeah, I don't know exactly.
Yeah. So I think this is, as we know, there is already a skin atlas bio-network in HCA. And this is, again, we can tap into that community and say, what are the standing metadata they're using? Because they're using that for Chloe's CAP, right? So there's already precedence to creating such effort. But now, because we're entering into disease and also spatial, right, we may need to expand that metadata. So we need to think about what needs to be added. Yes, sure.
In what diseases we have to collect microbiome data? More.
Get microphone.
In our data set at HS, we have the microbiome informations. But personally, I think that we have to collect the microbiome data if the disease, the hair follicular structures were involved in the disease pathogenesis. So that is my kind of the strategies that after I heard the other expertise opinions. So because of the skin microbiome, we haven't talked about the skin microbiome at all. So do you have any?
So I'm kind of speaking from the other side.
as a microbiome researcher. Like, completely agree. Like, I guess like as microbiome researcher, we like to say like microbiome is part of us. Like we have like what, like 100 times more gene. Like we say that all the time, but like it's not usually functionally the case, right? We don't have like 100 times functional capacity in the microbiome versus the host. So to your point, like it's really clear to like define the boundary, right? Like when do we need to profile the microbe versus the host? I think like one clear example is like when it's really involved in like host microbiome crosstalk, right? Through the immune system or like metabolite, let's say. So like I think in disease like Crohn's disease, like where I mostly do my research in, like it has like clear precedence or like implication of host and microbe coordinate like these like kind of like overt like inflammatory responses. So like I guess similarly in like maybe atopic dermatitis, I know a lot less about the skin, but like I feel like some work has to be done to like define like when is microbe needed, right? Because current methods, like we don't have like really sensitive methods for detecting microbes. Like in Physium, let's say, like we would detect because like bacterial RNA are not like polyadenylated, right? So it's an obvious challenge in some early technologies like Physium or like 10x. We've tried that like throughout like past technologies, but yeah, StereoSeq is like, I'm actually really excited about it because it's total RNA sequencing, right? But so there are like, I think technical hurdles we have to overcome, like microbiome, it's more like a 2D phenomenon, right? Rather than a 3D. It adheres to the surface and there's a lot less mRNA. Like there's what, like 300 mRNA per cell and like 99% of them are like ribosomal RNA rather than like actively like transcribing like messenger RNAs, right? So it's like a huge challenge. Like, should we like specifically do like a microbe-centric with hosts kind of atlas, like with like special design, or like should we leverage like these tools to kind of get it for free? I think these are a lot of questions and also a lot of challenges to think along the way.
Sorry, I put on the technical part. So especially sequence for, so I only had one paper about the target sequencing regarding the microbiome in the human samples during COVID. So what I think it's just like you can use a lot of target sequencing for, first of all, you can define some reference genes, right? You can pull out all the human like contamination, right? First. Then you can specifically to amplify the gene you're interested, right? As that can be taking 18 or 24 hours to the preparation, right? That's the first technical thing we did. Then for another things, I did talk to someone who actually do this. They said if they can identify the species of the microbiome, that'd be very beneficial as well. So hence, you don't need really sequence the RNA. You can just get the entire genome out and identify what kind of combination of the microbiome there. So each of the skins under each conditions, they have different kind of environment for different kinds of microbiomes. So it's not my area, but we did this for COVID to identify the COVID mutations, etc. So you're collecting different samples from different tissues, and the COVID will sort of like mutate differently. They have different pressures. So it could be applied to your skin stuff. So if you go and check the target sequence, to just design the probes that can be useful for yours. Because microbes, they express a certain gene. The operating frame is very small and the gene number is all defined. If you can identify the species, you might guess the function of it, right? All right, thank you.
It's like one clear genome, right? Usually very small genome like COVID or SARS-CoV-2, right? So people have done that and show like the more viral load you detect in like single cell RNA, like it correlates with clinical outcome, right? And say COVID. So like that's like, I feel like it's a really clever use case of like single cell it's designed for hosts, but like you also get like the viral like reads for free. But also, like to your point, like in a microbiome setting where we are like the host interacting with like hopefully a healthy commensal community, right? There's probably less species on the skin, but like in my application, like where we have like hundreds of species, then I think it's a different magnitude of scale to apply these technologies. So I think it varies across scale. Like you also have to think about contamination, as you said. Like we have like several problems along the way, right? Not just single cell. Like microbiome science, like there were like high level retraction of like some Nature paper using like TCGA, like to find microbial reads, and there were like lots of troubles. So yeah, I feel like if the human cell atlas, like I think I'm totally on board on like in relevant diseases or like say like AD or Crohn's, like I feel like if the technology allows, like you absolutely should like profile the gut the microbiome as well. But is it worth the technology, like how many years do we have to wait for the technology to mature?
Yeah.
Yeah. So like that's my main like concern.
Excellent. Thank you. Yeah. You just want to be mindful. So maybe 5 minutes, and then we need to close.
As a clinicians, can we change our microbiome? It's possible. I mean, for the therapeutic point of view, so scientifically, I'm not an expertise in this field, but the microbiome was established immediately after the birth, and then within kind of several months, they were established in our body. But some experts said that they think that they can change our microbiome, then that they will treat the disease. So is it possible? Like, is there any?
I don't think it's possible. Right? It's not like when you have the wrong kind of microbe, it makes you like sick. Like there's like host level adaptation. Like it is multifaceted. Like I don't think it's that straightforward. But at least I think, let's say in a skin microbiome based effort, right? Like identifying like what the say like strain genotype that correlates with disease activity. I think that's a good start. But yeah, like there are people who say like probiotics, like to reverse like disease causes. I think that's less supported by science.
Okay. Then maybe a last point.
Yeah. So actually, I can comment on your question regarding whether we can treat microbiome then treat certain disease. So I'm working on healthy aging. So there was one publication, they transplant, okay, as we age, the gut microbiome also age. So they transplant the microbiome of the gut microbiome of the aged mice to the younger mice. Then the younger mice become accelerated aging. Then they do vice versa. They say like, okay, the aged microbiome can be younger after transplanting with the younger microbiome. So to answer your question, yes, it can do, but depending on what kind of disease or what kind of health status you want to look at.
The application of FFT, but like say a more complicated disease where we would really benefit from creating atlases, right?
Yeah.
Yeah, we really have to define like when does the microbe contribute significantly.
Yeah, I think we're still doing trying to get more understanding regarding the host microbiome interaction. But talking about the FMT, I think safety is always a concern, whether we can find a healthy donor and whether the microbiome can be applicable to the whole population or not. That's one concern. That's why people are working on, for example, looking at the postbiotics, the metabolites from the microbiome or components from the microbiome, which can be safer and also stable.
Yeah. And I think it's in the realm of the skin. Yeah, I think there's also, because it's external, it's superficial, there's also an external environmental factor that also be more dynamic, which may be more challenging, but something for us to think about. I think whether we may not necessarily have to co-profile the spatial and microbiome, we can do it using two different technology. In theory, you do a quick tape before you do a biopsy, for example, and run separate. So there are various ways I think that we can consider the experimental design, but how to standardize. So, I mean, I can go right now to my lab and do it, but I don't want to do it and you guys not do it. Because I do it and you do it and you do it. We all do it the same way or more standard way. That's where the power comes. So hopefully through today's discussion, we have some. Yes, exactly. And I think key here is metadata, because once we have that template, then I can share that template with you guys, and we can all try to fill in the metadata as much as possible. So I think that kind of discussion will be needed going forward. So what I can do is to reach out to the global HCA Skin Atlas guys and say, hey, what kind of metadata do you have? Can you share that with us? And I can share that with you guys and see how we can build upon that. And that could be the first reasonable step that we can take. In terms of, we didn't really get to talk about different types of technology, spatial technology, single cells. I think we need another hour for that. So we won't continue, but just to let you know that there are different flavors of spatial. Let's see how we can go forward in the next meeting. Anything else that we want to mention? Well, Hyunje.
In our group, that we also have the scleroderma, the sequencing data. So we are open to sharing all the data to the old world. So just please let me know, and then we will sharing all of our data to everybody in the world.
Yeah, I think sharing data is another key aspect. Not only raw data, but also the expression matrix, but also sharing of metadata. Because some metadata includes clinical data, and so there needs to be some boundaries of what can be shared. So we should definitely talk about that next time. But I understand some countries, it's very difficult to share raw data. Can I just get a show of hands? Can you guys distribute skin FFPE sections out of your country?
Not for China, sorry.
So you cannot ship it outside the country?
Very restricted.
Yeah, Korea?
Even not to Hong Kong.
Okay, yeah, just cross to Shenzhen and collect. I see. Yeah, I understand, because we're also talking about how to create a hub so that we can generate data in a centralized location, but I understand that could be also difficult. So then sharing raw data of
US can't share any data if produced from BGI sequencer or
yeah,
Chinese sequencer.
yeah.
So any US collaborators, if you're using Chinese sequencer, you have to be careful. US.
Yes. I won't go into the details of that, but you're absolutely right. There's some caveats there. And what if you use probe-based spatial method? Then you don't have FASTQ file. You actually have image file. So it's not identifiable. That patient is not identifiable. Because the risk of raw sequencing data is you can figure out the SNPs and you can figure out who they are, which is high risk, and thus you don't want to share that. But image files like Cosmix or Xenium, it's image-based. Probably there's lower barrier to data sharing, so something for us to think about for the community. Okay, with that, I love the discussion. I think I heard yesterday they had a breakout discussion, 100 people, and nobody talked. So I think this is good. I think it's good size, and I think it's been very interactive. So thank you for your participation.